Hi, I'm David and this is the Biology Classroom. This is a paper discussion video, so get ready with this paper and let's go through the questions and answers together. Figure 1.1 shows the appearance of a Plasmodium-infected red blood cell. A, with reference to Figure 1.1, suggests how the presence of Plasmodium falciparum affects a red blood cell. Judging from the outer line of the cell surface membrane, the cell has lost its biconcave shape and is irregular. The cell surface membrane may have bursts or lice, hence the escaping daughter cells. A cell dies when the cell surface membrane bursts. You can also use the word hemolysis to describe the bursting of a red blood cell. We can see that many daughter cells of the pathogen have filled the cell. Red blood cells should have a darker shade under the electron microscope due to the presence of hemoglobin. The cell in the photomicrograph looks transparent, indicating that it has lost its hemoglobin. The pathogens may have used it up. Without hemoglobin, it can no longer transport oxygen. Some other logical suggestions include that the pathogen releasing toxins, and biological molecules inside the cell, such as amino acids, may be used by the pathogen to synthesize its substance. B. Outline the events that occur in the cell of Plasmodium falciparum to form the daughter cells shown in Figure 1.1. Plasmodium undergoes asexual reproduction to form new cells. They reproduce through multiple fissions, a unique form of mitosis. So, the events are similar to what you have learned about the mitotic cell cycle. First, they need to grow and synthesize more biological substances. Its cytoplasm increases in volume and mass. More organelles have to be produced to provide all the new cells. Then, DNA replication occurs. It is replicated in a semi-conservative manner. Now, the cell is ready for the nuclear division by mitosis. Unlike typical mitosis, they produce many nuclei because they must form more than 2 daughter cells later. When it is done, cytokinesis takes place. It is the division of the cell surface membrane and the cytoplasm. You can see in the photo that the daughter cells are budding off from the parent. C. Explain how antibodies will reduce the spread of malaria pathogen through the bloodstream. They are non-self antigens on the surface of the pathogen. Antibodies bind to these antigens as their antigen binding site has a complementary shape to the antigens. Note that you cannot call the binding site a receptor. Following the binding, pathogens in the infective stage are prevented from entering their target cells, red blood cells and liver cells. This is because the parasites use these ligands on their surface to bind to receptors on target cells to invade them. Antibodies also facilitate the destruction of Plasmodium by phagocytosis. They bind to mark the pathogen in a process called opsonization. This makes the pathogen more susceptible to phagocytosis. The binding also leads to the lysis of the pathogens. There is a mark for AVP. You can talk about how the binding enhances phagocytosis or the process of agglutination. Agglutination refers to the clumping of particles together. This reduces the number of circulating pathogens, which decreases their spread in the body. D. Discuss the factors that contribute to the success of a vaccination program. A vaccination program is more successful when it can access more people in the population. Herd immunity can be achieved when a large proportion of the population receives it. The remaining people who are not vaccinated can be protected as the disease will not spread among those who are immunized. Vaccinating children early enough in their lives can provide early protection, reducing the risk of infection. If the community are better educated about the importance of vaccination, more people will take part in the program. Government and NGOs with a good strategy for countering anti-vaccination campaigns and misinformation about vaccines will contribute to greater success. They can employ mainstream media and social media to achieve this. A vaccine that has a long duration of protection will be more successful as repeated vaccination is not needed. If the pathogen does not mutate, the same vaccine is effective all the time. Even if they do, The vaccine can still be effective if we can change and improve it in response to the changing strains. A plentiful supply of vaccines can ensure more people are vaccinated in a short time. We need sufficiently trained personnel to deliver the vaccine. The cost of production and transportation, the storage conditions, and the effectiveness in delivery to the population also affect this. They allow better distribution of the vaccine. Those vaccines with a long shelf life can be stored and distributed more easily, leading to a greater number of people being vaccinated. A stable vaccine will lead to a more successful program. They can be stored at higher temperatures, so transportation of the vaccine is easier. Lastly, a vaccine that does not require a booster would contribute to the success of the program, as a single-dose vaccine simplifies distribution, record keeping, and receiver compliance. Question 2 shows an enzymatic reaction that synthesizes amylose. Figure 2.1 shows the result of an experiment to investigate the reaction. The mixture is tested with an iodine solution, and the absorbance is read by using a calorimeter at fixed time intervals. A1. Explain why the absorbance increases. The mixture contains the substrate G1P and the enzyme starch phosphorylase. The enzyme converts the substrate into amylose. Over time, more starch reacts with the iodine solution and turns darker. The iodine solution changes from yellow-brown to blue-black when it reacts with starch. 2. Predict the results for absorbance if the students continue to take samples for a further 10 minutes. There are two possibilities. First, the absorbance remains constant. The graph levels off at 1.8. This is because all substrates are used up. The maximum concentration of amylose has been produced. In other words, the substrate concentration limits the reaction. It may also be due to the limit of calorimeter at around 2.0. The second possibility is that the absorbance continues to increase but less steeply, as the reaction is not completed yet. The increase slows down because most substrates have reacted. Even though the reaction is not complete, the substrate concentration has become limiting. 3. State an advantage of using a calorimeter in determining the progress of the reaction. The calorimeter gives quantitative or numerical results. This provides greater accuracy. These readings can be used to plot graphs. It is used for the calculation of the rate of the reaction. The numerical data also allows the use of a calibration curve to determine the concentrations of starch. The student can interpolate to estimate the concentration of starch in a sample. Comparatively, observation of colors by eye is very subjective. Judgments by our eyes are more prone to errors. In some cases, we can leave the solution in the calorimeter and take continuous readings. We do not have to take samples at fixed time intervals for the test. B. Describe the sequence of events that occur when starch phosphorylase catalyzes the addition of a molecule of glucose to the end of amylose molecule. Like any enzymatic reaction, the substrate will bind to the active site of the enzyme. They have complementary shapes. G1P and one end of amylose bind to the active site and form an enzyme-substrate complex. The active site changes its shape when substrates bind to fit better with them. This is described in the induced fit hypothesis. Enzyme decreases the activation energy. The enzyme provides an alternative pathway to the reaction to occur. A glycosidic bond will form between them. This reaction is condensation. A molecule of water is formed in the process. There is a mark for AVP. For example, you can talk about the formation of temporary hydrogen bonds between the substrates and the enzyme for the ES complex to form. You can also provide a detail of the nature of this specific reaction. Question 3 shows a hemoglobin molecule. There are 4 polypeptides in a molecule. 2 are the alpha globin and another 2 are the beta globin. P is the non-protein group found in hemoglobin. It is a prosthetic group called the hem. The diagram does not show the primary structure, as we cannot see the sequence of amino acids in the polypeptide. The secondary structure is shown. As we can see the alpha helices. It is a three-dimensional image, so the tertiary structure is shown here too. There are 4 polypeptides in the diagram. This is the quaternary structure. B. Explain how hemoglobin is involved in the transport of carbon dioxide molecules that are not converted to carbonic acid. Hemoglobin can combine directly with carbon dioxide to transport it. However, carbon dioxide does not bind to the hem group like oxygen does. It reacts with the terminal amide groups of each polypeptide. So, a molecule of hemoglobin can transport 4 carbon dioxide using this method. The reaction forms carbaminohemoglobin. Carbon dioxide is bound to the hemoglobin until it reaches a region with low pCO2. In other words, it is bound until the red blood cells arrives at the pulmonary circulation. Due to the breathing process, carbon dioxide is constantly removed and oxygen is taken into the lungs. This causes a reverse reaction where carbon dioxide is released from the hemoglobin. Hemoglobin molecules bind with oxygen at the capillaries around alveoli in the lungs. This is where the PO2 is high. D shows the oxygen dissociation curves when red blood cells have high and low concentrations of 2,3-DPG. One wants you to describe the effect of an increase in the concentration of 2,3-DPG on the oxygen dissociation curve. The curve shifts to the right at a higher concentration of 2,3-DPG. This means hemoglobin has a lower affinity for oxygen. We can tell by comparing the percentage saturation of the two conditions at the same partial pressure of oxygen. You can see that hemoglobin in this situation is less saturated at any given PO2. In other words, a higher PO2 is required to reach the same percentage saturation as the low DPG concentration condition. P50 is higher too. You can use the data in the graph to get this point. 2. State and explain the effect that the use of blood taken from a blood bank has on the supply of oxygen to the tissues of a person during an operation. The graph shows that the affinity of hemoglobin to oxygen is higher when there is a lower concentration of DPG as the curve shifts to the left. A higher affinity means that hemoglobin has a greater ability to bind oxygen. It does not favor the unloading of oxygen. This causes less oxygen dissociation. Less oxygen is released and becomes available for tissues. In question 4, we have a diagram showing the transcription of a gene. The enzyme involved in this process is the RNA polymerase. It catalyzes the formation of bonds between the free activated RNA nucleotides to produce the mRNA. The bond is called the phosphodiester bond. B1 wants you to state the number of amino acids that are coded for by the sequence of nucleotides on the primary transcript. Every 3 bases is a codon that codes for an amino acid. The primary transcript has 16 nucleotides so far, so it codes for 5 amino acids at this point. Using the information in figure 4.1 to explain why one of the strands of DNA is not transcribed. In transcription, only the template strand is transcribed. The other strand is not the template strand. The sequence of bases is used in translation to determine the sequence of amino acids in the polypeptide. So, if the non-template strand is transcribed, the sequence of nucleotides or bases in mRNA will not be the same as the intended code. It will code for a different sequence of amino acids. This changes the polypeptide's primary structure, which in turn alters the protein's tertiary structure, resulting in a non-functioning polypeptide. The production of a non-functional protein would be a waste of energy. Besides, the first codon of this mRNA would not be the start codon that signals the initiation of translation. Any suggestion of a possible consequence to the structure of collagen can give you a mark. For example, one of the codons in the middle may be a stop codon. This causes a shortened polypeptide to be produced. Another reason is that mRNA is single-stranded, so we do not have to copy both strands to produce a double-stranded molecule. Lastly, RNA polymerase can only synthesize RNA in the 5' to 3' direction. the non-template strand would be in the opposite direction as the DNA strands are anti-parallel. In C, we have an electron micrograph of a dividing mitochondrion. 1. State two features of mitochondria that are visible in figure 4.2. The two outer lines show the double membrane of the organelle. It is called an envelope. The projections pointing to the center are the folded inner membrane. They form cristae. Those small faded dots inside are the ribosomes. The matrix can also be seen. It is the gray fluid-filled part in the center of the mitochondrion. 2. Calculate the actual diameter of the mitochondrion. The actual diameter of a structure is the image size in the photo divided by the magnification. Use a ruler to measure the image diameter in millimeter. Then convert it to micrometer by multiplying by 1000. Dividing the value by 50,000 will give you the actual size. D. Outline the ways in which ions can move into mitochondria. Mitochondria are membrane-bound organelles. The way ions can move into it is similar to how they can enter a cell through the cell surface membrane. Ions are charged, so they cannot pass through the hydrophobic region of the membrane by diffusion. They must move through by active transport or facilitated diffusion. Active transport uses ATP, and the ions move against their concentration gradient. Facilitated diffusion is a passive movement. It does not require energy, and the ions move down their concentration gradient. Both processes may involve carrier proteins. These transport proteins can change their shapes. They have specific binding sites for the substances they transport. Facilitated diffusion can occur via a channel protein or a pore protein too. These transport proteins do not undergo conformational changes. They provide hydrophilic pathways for ions to move through. There is a mark for AVP. For example, you can talk about the hydrophilic pores in channel proteins or the specificity of carrier proteins. A1. Explain why a root can be a source and a sink. A source is a site in a plant which provides organic compounds for other parts of the plant. Sources are typically photosynthetically active parts of the plant. A sink is a site in a plant that receives organic compounds from another part. The root commonly acts as a sink because it is a storage organ. It receives assimilates such as sucrose and amino acids from the source to store and grow. Even though the root is not a photosynthetic part, it can sometimes act as a source. In some circumstances, it can provide assimilates for other parts of the plants. For example, when a plant needs to use stored energy in the roots for new growth as the leaves are not photosynthesizing. Due to the bidirectional transport in the phloem, the movement of assimilates does not only occur in a single direction. The direction of movement can change with the demands of plants for assimilates. So, a plant organ can be a source and a sink in different conditions. Sucrose and amino acids are two commonly transported assimilates in the plant. B. Describe and explain the mechanism that is responsible for the movement of phloem sap in sieve tube. Assimilates such as sucrose is loaded into the sieve tube elements from companion cell before it is transported inside. They diffuse into phloem sieve tubes from companion cells via plasmodesmata. Plasmodesmata are microscopic channels that connect the cytoplasm of the cells. Assimilates are soluble in water. Their presence in the sieve tube elements causes water potential to decrease. This leads to the movement of water from surrounding tissues by osmosis down the water potential gradient. The entrance of water causes the hydrostatic pressure to build up. Hydrostatic pressure refers to the pressure exerted by a fluid in a confined space. At the sink, the unloading of assimilates occurs. This lowers the hydrostatic pressure. There will be a pressure gradient due to the difference in pressure in the sieve tube elements near the source and the sink. The pressure difference leads to mass flow. Mass flow is the movement of fluids or substances in bulk down a pressure gradient. This is because, as assimilates move out sieve tubes near the sink, water potential increases. Water will then move out down the water potential gradient. C talks about a virus called TMV that infects crop plants. Figure 5.1 shows the accumulation of TMV in leaves for 3 varieties of tobacco plants, C, V1, and V2. The question wants you to compare the results for variety V1 and V2 with the control groups of plants, C. The command word compare means that you must talk about both the similarities and the differences. For similarities, you can say that all varieties have the same pattern or trend. V1 and V2 can reach maximum accumulation, just like C. However, the maximum accumulation is delayed in V1 and V2. They do not achieve it as quick as C. Quoting data to support this point will give you another mark. The rate at which accumulation increases for V1 and V2 is lower than the control. This can be determined using the steepness of the graphs. Since both V1 and V2, which have lower quantities of PME, can still reach the maximum accumulation, a low quantity of PME does not affect the maximum TMV accumulation. 6a wants you to explain why the ventricles have thicker walls than the atria. There are 3 key points to include in this question: distance, pressure, and resistance to the blood flow. Ventricles pump blood to a greater distance. The right ventricle pumps blood into the pulmonary circulation, and the left ventricle pumps blood to the systemic circulation. The atria only need to pump blood into the ventricles. To allow blood to move for this great distance, ventricles have more cardiac muscles to pump blood at a higher pressure. They push blood into the aorta and pulmonary artery with more force than the atria. This high pressure allows the blood to overcome a higher resistance, allowing it to reach the intended destination in the body, which may be far away from the heart. B. Complete Table 6.1 to identify the structure in the heart responsible for each function listed. The sinoatrial node initiates the heartbeat by sending out a wave of excitation to the atrial walls. The impulse cannot reach the ventricles directly, as there is non-conducting tissue between the atria and the ventricles. AVN is stimulated to relay the impulse to the bundle of His. This causes a delay between the depolarization. Purkinje tissue or Purkinje fibers are embedded in the ventricular walls. They transmit the wave of depolarization through the wall. When the left ventricle contracts, the left atrioventricular valve, called the bicuspid valve, closes to prevent the backflow of blood into the left atrium. That's all for today. If you think my videos are useful, don't forget to like, comment, and subscribe. Thank you for watching, and see you again soon.